Welcome back, guys. Today we are on immune system part 1. There's a ton of information for this, so it is split into 2 sections. And I just want to start with a little table of something that is covered in the pre-lecture assignment, but we don't really get into in the notes. And that is this concept here. Compare and contrast innate immunity and adaptive immunity. So just up here, we're going to make a little table. So innate and adaptive. So we're going to split them in half here, and we need 3 little rows. So innate immunity is nonspecific. So it doesn't fight against specific infections, whereas adaptive does. Innate immunity kicks in immediately after infection. And adaptive is going to be anything that gets past this innate response. And like it talks about in the pre-lecture, the innate is things like your, what's the analogy for this? This is like protecting your house. So imagine that your body is your house. Your innate immunity is just non-specific things you're going to do to protect your house from invaders. So what would you do? You could put up a fence. You can lock your doors. You can put up cameras. You're not protecting your house from anything, any type of specific criminal. You're protecting your house from being invaded in general. So non-specific happens immediately after infection, and it has no memory of past infections. So with adaptive immunity, this is anything that's going to get past those barriers. It gets past the fences, it gets past your cameras that can unlock your doors, and then the adaptive kicks in. So think of things that you would have in your house to protect against specific type of invaders. Maybe there's someone that's after your money, so you would want a safe to keep that money safe. Or maybe they're violent intruders and you would want weapons to protect yourself against them. What's another example? That's all I can think of right now, but just specific things that your body does to protect from a specific type of invader. And these ones do have memory of past infections. So just make sure you can compare and contrast those two. So I don't remember if I said this already, but this is things like your skin, the mucus lining your nose and your airways, just things that are going to try to stop invaders from even getting in initially. So for our question of the day, how are antibodies used in medicine? Which, as always, we'll get to later. We're not going to go through any of this today. We're going to jump straight into innate or non-specific immunity. So remember, this is your first line of defense, kind of like your fences, locking your doors, having cameras at your front door, relating this back to your house. So this causes an inflammatory response. So the second line of defense, the first line would be physical and those chemical barriers. This is the second line. So histamine is released from damaged tissues or from something called mast cells. And what histamines do is they increase blood flow. And when you do this, it's going to bring heat to the area as well as swelling, and it can be painful. It also causes capillaries to be more permeable to white blood cells. Remember how the white blood cells can kind of sneak out into the tissues through the capillaries? So it causes capillaries to be more permeable to white blood cells. That increased blood flow is going to bring macrophages to the site. And remember, macrophage means big eater. So these are going to engulf or eat the invaders. And release something called cytokines. And a cytokine is, you can think of it as a chemical call for help. Like, hey, we've got some invaders going on. We need some backup. So it's like summoning help. You can also think of it as being like a flare gun. Hey, we're over here. We need some help. Got some great acting skills there. Also, you're going to have neutrophils and monocytes arrive. So the monocytes will mature into macrophages. Which remember engulf invaders and release cytokines. And then both neutrophils and monocytes engulf pathogens. And I don't know if you've heard the word pathogen or if we've defined it, but it's just a something that can get into the body that can cause disease. So viruses, bacteria, anything like that. And they release cytokines as needed. So let's see if we can zoom in on this picture here. No, my camera doesn't do the greatest job of this. And this is tiny print. It's even blurry for me looking at it just on my page. So anyway, we've got a capillary here. And let's say we got stabbed with the tip of a knife and it went into our capillary. So injured tissue cells, oh, sorry, this is a capillary here. Injured tissue cells and mast cells release histamine, which causes capillaries to dilate and increases blood flow. So we've got our blood clot going on here with that fibrin tissue with the platelets. And then the macrophages phagocytize, they eat the pathogens, release cytokines, which are these little blue dots here, which will stimulate the inflammatory response. You'll get neutrophils and monocytes becoming macrophages, and they squeeze through the capillary walls out into the tissue and gobble up those pathogens, the green guys. And then back to the blood clot. Blood clotting walls off capillary and prevents blood loss. Okay, so histamine is released. This is going to increase blood flow, cause the capillaries to be more permeable to those white blood cells. It's going to bring in more macrophages so they can engulf the invaders. That will cause them to release cytokines, kind of like setting off a flare gun. You also have the neutrophils and monocytes engulfing pathogens and releasing cytokines. You also, this might be better close up like that, huh? Let's try it out. You also have protective proteins. So several proteins just circulating in the blood aid in innate immunity, and two examples of these proteins are complement proteins and something called interferons. So what complement proteins do is what you're seeing in this picture over here. So they will bind to bacteria, poke holes in it, and then because of osmosis, fluids rush in, that bacteria swells, it pops, and it's dead. So these little tiny red guys are the complement proteins. This is a bacteria. It's going to bind to it in this little circle, pop a hole in it, or let the fluids in, and then it bursts, it dies. So know that that's what a complement protein does. And the interferons are proteins released by virus-infected cells. And these will bind to receptors of non-infected cells, and they signal non-infected cells to vamp up antiviral defenses. So this is kind of like if your neighbor gets robbed, they're going to come to your house and say, hey, guess what just happened? Make sure you're locking your doors, make sure you're prepared and paying extra attention to your surroundings and watching out for bad guys. That's like what the interferons do. So make sure you know that as well. So I'm just going to write over here, warn the neighbors. This is a little too close for me. Let's see. There we go. Okay. All right, so that was our innate immunity, which is nonspecific, and now we're going to move on to adaptive immunity, or specific immunity. So this fights infection that spreads past that innate immunity, and it targets specific pathogens and remembers past exposures, like we talked about on the very first page. So this is not an immediate response because it needs time to build up these cells. And so first it has to recognize and identify antigens. And just to review, antigens are protein markers that identify something as being foreign. And so back to what we talked about, I believe it was it was yesterday for me, last time for you. As cancer cells mutate, they start to develop antigens that are no longer identified as self, but at this time they're usually far enough into their development or in the later stages, and that's when your immune system starts to recognize, hey, these are not normal cells. These antigens are found on fragments of bacteria, viruses, cancer, etc. And antigens distinguish self from non-self. So this adaptive specific immunity is carried out by lymphocytes, which are your B and T cells. So they can recognize antigens, and some can retain memory of past infections. So B cell lymphocytes are what go through antibody-mediated immunity, and then there's also one called cell-mediated immunity. So each B cell contains a unique antigen receptor. So here are some B cells hanging out, and they all have these unique shapes on their antigen receptors. And these look like antibodies, which can be confusing. They're not. These are just receptors on the surface of the B cells. And there are, I forget the number. I think the number is about 2 million of these different receptors just hanging out in your body. And if you have an antigen here that matches up with a specific receptor, activation will occur, and you will get something called clonal expansion upon recognizing that antigen. So the B cell that found the matching antigen will multiply. It will clone itself and expand throughout the body, clonal expansion, because that's what we need to help us fight the infection. Oh, and it does say here on my notes, 2 million, I was right. 2 million different receptors in your body. So if one of those gets activated, triggered, it can bind, and fits with one of these receptors. Clonal expansion will occur. It'll make a whole bunch of copies of itself. And this will cause amplified B cells to mature into either plasma cells or memory B cells. So plasma cells have lots of rough ER. So that's going way back to our cells chapter. The rough ER, there's rough and smooth, and the rough is kind of like studded with ribosomes, and ribosomes make protein, and antibodies are proteins. So it makes sense that they would have a lot of rough ER. And these plasma cells are going to make and secrete antibodies that match the antigen. And then when the infection ends, they undergo apoptosis. Remember, that's programmed cell death because we made all these copies of them, and when the infection is gone, we no longer need them. So they'll go through apoptosis. But the memory B cells will stay, some of them will stay in the body to remember the antigen. That way, if the antigen is encountered in the future, it will launch a faster, stronger, longer lasting attack. Because it remembers, it's ready, it's specific. So over here, along with the clonal expansion, we are getting those plasma cells, which will secrete these antibodies here that match the antigen. And then when we're all done, they can go bye-bye. But the memory B cells, like their name says, they remember. So let's talk about antibodies. These are Y-shaped proteins. So we're zoomed in on one here. And they have a variable region that binds to the antigen. So right here, it's like the tips of the Ys. It's the region that is going to match up to a specific antigen. And once they bind to the antigen, they can recruit complement proteins. And remember, those are the ones that poke holes in the bacteria and they cause them to fill with fluid and explode and die. They also trigger phagocytosis, which is cell eating. So anything that's covered in antibodies, the macrophages just want to come eat it up. That's like a symbol like, hey, this needs to go. They're going to come gobble it up. They can prevent pathogen function. So COVID-19, for example, I know we're so probably overhearing about this, but it's a coronavirus. So it has these spike proteins on it that are kind of shaped like this, that go all the way around the whole cell in 3D, not just like I'm showing it in 2D. And so if we have an antibody that can come and block all of those spike proteins, it is going to allow it to not be able to attach to the human cells, and it won't be able to make you sick. It also can inhibit toxins. So there are some bacteria that release toxins into your body, and it can prevent them from affecting you as well. So our question of the day, how are these antibodies used in medicine? These can occur naturally, but they can be made in a lab against a specific pathogen. So monoclonal antibodies are antibodies that originate from a single B cell, and they can bind to one specific antigen. So if you think about the possibilities of this, if we can make these antibodies in a lab that are specific for one type of antigen and then give them to people, I don't know in which way, if they inject them or take them. Looks like this one is an oral medication. Oh no, that's an HIV test, my bad. But yeah, they can be manufactured for medical use. So right here, this is showing some of us may recognize this, a pregnancy test. And the way that the pregnancy test is actually used, actually works by using antibodies. And I didn't know this, but what a pregnancy test is detecting is something called HCG, which stands for, it's been a while, human chorionic gonadotropin, which is something that is found in the blood of pregnant women after a certain amount of weeks of pregnancy have gone by. And so with an HCG pregnancy test, the P stick, I'm just copying my notes here, the P stick, the urine indicator, sounds a little more professional, has antibodies that bind to HCG and this will cause a color change. And depending on the test you have, you'll get a double line if you're pregnant, because it'll cause a color change in one of those lines. Sometimes it's a smiley face. Sometimes I took digital ones that would say like pregnant or not pregnant. I'm not sure how that works, but they all have antibodies that bind to HCG, and that's how it causes some sort of change in the indicator that lets you know whether or not you are pregnant. So that's pretty cool. I didn't know that. Diagnostic viral testing. So again, it can test for the HCG hormone. And it can also be used for HIV testing. So there are at-home kits like this where you can, I believe, you swab your gum with this one. And then they have antibodies that recognize the HIV virus. And if you have it, it'll cause a color change, and that's how you can see. For cancer treatments, a couple of things, they can be linked to radioisotopes. So when they reach cancer cells, they glow, and you can see this sometimes in a PET scan, but just you don't have to know the details, just that it can be used for cancer treatments. And it can also produce cancer antigens. I feel like I'm going too fast again. I'll pause for a second and just talk. So it can also be used for COVID-19 treatment. So something called Regeneron is where they, in a lab, produce these antibodies specific for COVID-19, and so they could give that to you as treatment to help your body fight off the virus. All right, before we move to T lymphocytes, which is going to be next, let's look at this flow chart. And it looks pretty wild. Don't let it freak you out. It's fine. But we already talked about B cells, so we're going to go ahead and fill in the B cell portion of this because it's combining B cell and T cell responses. So the B cell starts right here. So remember with the B cell, the first thing that happens is the B cell binds to an antigen. And this will activate B cell clonal expansion, like we saw. It'll recognize, hey, we need some more of us to fight off this pathogen, so let's clone ourselves and expand throughout the body so we can help fight this. And we get plasma cells from this, which ultimately produce antibodies. And then we also get memory B cells that will remember the infection. So that's the B cell portion of it. The rest of this is going to be T cell, and you'll notice that T cell branches off to activate this B cell portion. So we will come back to that in a bit, but let's talk about T lymphocytes. And this is cell-mediated immunity. Doing the paper shuffle here. Lost my spot. Alright, so T cells have receptors but cannot directly recognize antigens like the B cells can. So there are 4 steps here. The macrophage, which is this guy, eats an invader. So here he is gobbling up a bacterium and displays an antigen to a T cell. So these are all T cells. And this one is one that matches up. So he displays it to the T cell. Think of the T cell as being like the king and the macrophage is like the servant. And he's like, here you are, sir. Here is your antigen. And so what that is going to do, the T cell will bind to that antigen if the receptors match. And then that T cell that it binds to is going to release those cytokines, which is like the chemical call for help. And this induces clonal expansion. So now we're going to make a whole bunch more of these specific T cells that we need. So this is going to produce a whole bunch of helper T cells. And then back to that flow chart, and we'll come back to that. B cell activation is dependent on those T cell cytokines. So this is going to activate B cell clonal expansion like we saw before. It's also going to activate cytotoxic T cells. And cyto is cell, toxic is poison. So these are like deadly T cells that bind to infected cells and induce cell death. And it's going to make memory T cells, which are going to remember the pathogen and jumpstart in case of future exposure. So we've got the macrophage. He gobbles up a bacterium. He presents it to Sir helper T cell, who binds with it. This causes it to release cytokines, causes clonal expansion. We get a whole bunch of copies of itself. We get cytotoxic T cells, which can bind to infected cells and induce death. And then you also get memory T cells that hang around and remember the pathogen for future exposure. That was a lot. I know this is a lot. The immune system is a rough one. So let's finish with our chart here. All right. So with T cell, you have that macrophage. That eats the invader. He gobbles them up and displays that antigen to Sir helper T cell. And then remember what happens then is Sir helper T cell secretes that chemical call for help, the cytokines, and this is going to activate the B line, which we already went through. Going the T direction, it's going to activate T cell clonal expansion, which does three things. You're going to get more helper T cells, which secrete cytokines, call for even more help. You're going to get cytotoxic T cells that kill already infected cells. And then you're going to get memory T cells to help remember the infection next time. Whew. So we're going to talk about this more next time, but the way that HIV works is it attacks right here, which is the very top of this whole cascade of events. So it's going to prevent this B cell from all this stuff happening. It's going to prevent all this T cell stuff from happening. And so you have no specific immunity because HIV stops everything happening right here. And that's why it's so serious. Okay, so this page right here, I would suggest, of course, just going over this a few times and then maybe making an extra copy of this, or even just drawing out what's going on your own. That would be a great way to study for this. And I know it's a lot, but you guys can do it. You've done great. I've looked at the results from exam 3. They are pretty good. I do have most missed questions on exam 2 ready to go, which was forever ago, and I'll try to do it for exam 3 as well. Um, this is just going so quick. I'm having a hard time even keeping up with being on time with the lecture, so I apologize for that. Um, yeah, let me know if you guys have questions, and have a great day, morning, night, whatever time it is for you. Bye.